All right, so good morning, everyone. I just mentioned to Lai, I don't know if everybody heard, but at the end of this session, I will go ahead and send both the one from Tuesday, the Zoom recording, and then the recording that we get whenever it converts and is ready for downloading on Teams. I'll send that at the end of the day for you guys to look at. And for today, we have, we're going to go over neuron tracing. So I have a couple of example data sets. We will start with looking at neurites, like just images of neurites, images of neurons, so having like cell bodies attached with them. And then I'm going to go and show you, I mentioned a little bit, we can do some reconstruction and deep learning to help kind of pull out some contrast and maybe fill in some regions on neurons. So I'll just kind of show you what I did to build a model like that, and then just applying it. So I'll go ahead and share my screen. We can start first with maybe this data set where it's just neurites. So I'm going to show it in 3D here. And you can see that it's just neurites. It has some spines coming off of it. Then there's no cell body there. And so a little bit of background, but it's not enough to really looks like it affects kind of the contrast or what we can see from this neurite. And you can see that it's pretty even in staining. Like there's very that's probably the most it dips and goes away in terms of intensity, but you can kind of make out for all of this that it's just nice and connected. So this is ideal. And when we go and we want to trace something like this, there's both an automatic tracing method and then a method that you can do it manually as well. And then kind of a combination of two. You can start with like an automatic tracer using the function that I'm about to show, and then you can edit it after the fact and correct for any mistakes if it's needed. So I'm hitting this analysis panel button right here. I'm going to hit new pipeline. Alternatively, you can also go down and do this detect neurites, but I'm just going to build it from scratch. So I'm hitting new pipeline, and first step is always taking this input ROI. I would have already imported this image. I didn't show you that process here, but this was, whether this be a CZI or TIFF or some other file, there's first step is always importing. And then once I've imported, I created a copy of this document as a .sis file. Now that I have it opened, I'll then input the area of the image that I want into my analysis pipeline. So in this case, I'm just going to analyze the whole thing. And then just to give you an idea of the size of it, it's a really small image. So it's only about 8 megabytes when it's compressed. It's an 8-bit image and some pixel size information. Can reduce this a little bit. So we're gonna run it on just the full image set because it's a really small image and it's not gonna take that much time. So hit run pipeline to here, and then whenever we go and we set up our pipeline, kind of figuring out settings. So I'll go into this add operation. Maybe I'll go in order first. So I guess the first thing when you're starting a pipeline is, if you remember from Tuesday, that we always have to kind of work in order. So the first step is image processing. So for this case, because the background is really low, or kind of is expected, you can make out the contrast between the background and these neurites, and there's really not a lot of regions. Maybe here it's a little cloudy, but the software, there's such high contrast that the software will be able to discriminate against it. So I don't think we have to do any pre-processing in this case to get a nice trace. I will show you later doing some image processing beforehand, but because we don't have to do any in this case, we're just going to skip the step and we're now going to go to the segmentation. And so for this, we would then go down and then we would find, maybe it would be in segmentation, or maybe it's in, no, it is. So You can either type it in here. So because I know this is nerd, I can just type it in, but I know that it's in segmentation. It's the last option. So I'm going to double click this. And there's a few options here. So one, there's the naming of the operator. You can always change this to whatever you want. So if you're working with maybe a specific like image file or data set, and you kind of want to keep it like some tag that's just really identifiable, you can either change it here or just change this down here. So, if you're working with like, I don't know, Neuron in like condition 1, something like that, I would rename it just so it's um, identifiable and it's just a good housekeeping rule. In this case, I'm just going to call it trace nerd or something basic like that. And then for the outputs, you also have the option for selecting how you want them to be output. So in this case, this is going to be the color of the object. Let me go ahead and I heard someone. In this traced neurite, the output is just going to be multicolor. So that means for every individual neurite that you have, it's going to have its own color associated with it. You can also use a fixed color, keep it random for now. And then you have this eyeball again for most of the operators. It works in 2D, and for a lot of the operators, one of the recent updates we just had, we can do it in 3D now. So if I hit this eyeball, you can see I can drag this bounding box to the length of my image. You can see that it's trying to make some guesses on where these nerds should be traced. And right now it's not looking good. I mean, it's getting this, but it's missing this entire branch, which is not what we want. And we also have a speed version of this and then accuracy. So accuracy will be a little more predictive of if we were to run it. And it actually looks a little bit better. Still missing some gaps here, some here. So we'll see if running this will fix it. And then in this hamburger menu right here, there's also a few more options. One of the things that I find useful sometimes is just running pipeline to here. So let's say you have like, I'm just going to create a bunch of operators for a second. Let's say I wanted to test a whole pipeline, but I'm not sure about maybe like this step right here. You can just run it, click a hamburger menu button on whatever operator you kind of want to run it to, hit run pipeline to here. It's kind of similar. If anybody does any coding in Python, you kind of put like a little stop mark at a certain point. This is kind of the same idea as that when you're testing and developing pipelines. Go ahead and delete these because we're not going to do any watershedding. I have the show preview button, and then you can also, for this case, it's pretty limited, but there's also other operators who will allow you to have more options in terms of like fine tuning some of the applications of the operator. So if you remember from Tuesday, we had that deep or cell pose that we applied. And with that cell pose that we applied, there was also like some smoothing operators or a smoothing value. There was also like a threshold value too. And then I think that's it for kind of this menu thing. And then, of course, if you have any questions or if you're just curious about how it operates, you can hit this help for any of the operators. The same thing is going into this help up here and going to either the manuals or this help button up here. But this will kind of bring you to where the operator is within this help system. And then for some of these, as I mentioned before, they'll have some papers on this. And they'll go a little bit more into kind of what the meaning of this is. For example, like a tubularity map. What does this mean? Etc. Right. So for here, I want to fill in some of these gaps, and probably the reason that it's stopping here is because there's this drop in intensity, and so that may be below this threshold that we have here. So I'm just going to kind of reduce this a little bit.
Brennan?
Yep.
I just wonder, like, currently now, what does that ball means here? If the shoes
What
are
does what does
the ball,
yes.
I mean, you can see that there's a line connecting those kind of blue balls.
Yep. So this would mean that it's essentially like a node, you can think of it. So if there's a case of like you would have your starting point, and then you would have these cases of where it's connecting each of these, like what it's considering these nodes together. So this would be, it's recognizing that there should maybe be a deviation. So this is like a spine that's coming off of it. And then it'll have like a ball, and then a line, which is where the neurite will be, and then another ball. To the end of this. And once this is just a really like a skeletonized version of the rendering. When we actually implement it, it's going to fill out to fill into the gaps of where the intensity is. So it should smooth out and fill up and take the entire like volume.
Okay.
And so I lowered a little bit. It kind of got there. A little bit more. A little bit better. And this is also just a kind of its best guess. So it's not actually implementing this math, so it may not be super accurate, but we'll see. Maybe we can make it look a little smaller. Yeah. Too much. Okay, maybe that's better. And then we'll see. If I run it here, it's going to go ahead and trace it. Close this out. Okay. And then we have what's its best guess right here. So if I go and zoom in, you can see that there's some spots that it's missing, but it seems to be getting like this one over. Let me go to this pan tool. You can see it's missing this. Oh. Does anybody have a question?
Lin, can you silence your microphone?
Oh. Yeah, there's no question. And then the couple areas that are missing, it's getting some of these. And what we can do is we can either do some adjustment with this. So if I'm not happy with this, just go ahead and kind of mess with it a little bit more. Maybe I can change this value to be a little bit longer. But the point being, what I'm doing here is just kind of taking my best guesses on what works and what doesn't work. There's not really, so in this case, it looks like I have more traces. That's probably not what I want to do. Maybe it is actually. So you can see it's filling with a little bit more sensitivity. It's filling in some of these other spine regions that were missed before. Maybe this is a good starting point. And then you can also see that there's like some other regions that we may or may not want. We can see that there's some, like for example here, we have really low settings, so maybe it picked up some of these other objects that are really low in background. Some here, some here. And I think there was one little spot over here that probably you would want this to be connecting, right? Okay, so I think this is a good starting point. And what we can now do, as I was saying before, we can kind of do some adjustment. And to do that, we can hit this trace tool right here. And so if I hit this, it'll now go back to that skeletonized version of what we saw when we were kind of setting up our settings here. And we have the option to kind of adjust and change any of these nodes as needed. And the way that we can do this is, let me start with this one. Can just take this and connect them here. Oops. So there's up here these few options. So we have these modes. This is just a selecting tool. So if you have this object table and you kind of want to click around, you should be able to click through these. I'd have to pull them up. You can click around on these nodes. but we'd actually want to go and draw. So we can, in this case, it's saying draw a new trace or continue an existing one. We also have the option to select or remove some of these branches, remove some of these nodes. So for example, if I wanted to get rid of this node, or we can get rid of the entire branch. So I can get rid of a node. It'll kind of break these back up. The entire branch. And then there's also a few other options here. So depending on if you are doing this automatic tracing, then go and do this. You can see that as I'm hitting this draw button, and then I'm starting with wherever node I kind of want to build off of. And you can see it's kind of snapping, right? Like it's kind of, it's not like a direct straight shot. I'm trying to mess it up a little bit. So here you can see it's just a straight line, and you have this little error right around where my cursor is that it's saying that it's too far away from the start to use this automatic mode. And part of this is just going to be, way too far away, and it doesn't have a path to kind of find it. It will try its best to make sense of a path. So you can see here, there's actually nothing that I can see obvious that is connecting these two, but it's using some, you can see it's kind of finding a spot here and kind of jumping between these. Let me let this person into. Jumping between these nodes to try and make sense on whatever this background is. So this can be really useful when you're going to trace, because let's say I did want this entire thing. I don't have to go and click each node one by one. I can just hit this. I started at this node, I hit the end. It kind of took a guess on where the rest should be. And then when I go and I do this, it's going to fill it in. Go back to this edit. And then you also have the option to, if you want to make this, have this option for a starting point, you can do this, make a primary branch. So if anybody is working with maybe trying to determine branch order, something like this could help if you want to start with like a primary branch and then kind of the dendrites that are spawning off of it. You can add some tags to this too. So this is going to be, are you trying to trace or trace like a cell body? In this case, we're not. So that's why we have neurite selected. But if we did have a cell body, I guess maybe we'll go in that in the other example. Neurite, one more time. Let me do this. And then there we go. And then you have a few other options here too, with kind of some of the same things that, if you just click, it'll end this trace here, or you can just hit accept once you're finished tracing. There is a way that you can also refine this connection. So if you have these two starting points, you can kind of use some automatic tracing again to re-estimate it. It is going to be, I would say that these two options here can be sometimes helpful. Like, for example, if you wanted to maybe simplify this connection here, actually, I can go and try it. I'm not sure how it's going to behave, but let's give it a shot. So maybe I wanted to kind of get rid of this connection to unable to connect. Let's see. And I wanted to, well, maybe not that one. Maybe let me find another connection that is more simplified. Let's just see. So you can pick these two starting points. I'm not sure if you guys saw it. Maybe I'll pick a better example, but you can see with this simplified connection, if I go back, redo this one more time. So you can see it has this shape here. If I hit this refine connection, you can see it kind of adjusts a little bit. So this is another way, instead of just having to drag and drop to kind of what I did up here and corrected it, you can just say, here's point A, here's point B, recalculate this tracing. It can be useful for some examples, but usually what I would recommend is, because this is still using some math and the local intensity, using its best guess to trace these, I would recommend just starting off when you're doing this automatic tracing using this function here, just get the best result that you can out of here. And then I'd probably just do some manual editing afterwards. You can try and see if doing this simplified connection or refining it can help, but in practice for me, it usually will just change a few, like the shape a little bit more, but the nodes aren't going to start connecting into regions that we wouldn't get if we didn't get it when we just implemented the function from the get-go. So we can see if that is true here. Maybe it will fix it. That's not what I wanted to do. Back to this tracing. Let's see if we can refine this. Select this trace. And if I go and hit now refine here, okay. Oh, okay, it did work. All right, sometimes it works. So in this case, it could be useful just to have, maybe a smaller region and then just have it refine it, because maybe it's now using, I'll have to look into the settings on what actually it's using to implement this refined connection, but it may be using like a local contrast in the region that we're focusing on. Can see if there's anything there. Out of the box. I'll have to look a little bit more into that. But yeah, I would probably just recommend doing it manually. So for example, here, find a little spot and then click, and it just does it automatically. Okay. So we connected this. All right. There is also, see here, that's editing trace. Okay, I think that covers these. Oh, maybe we can do splitting objects. So these objects, or these are now considered traces, and there is, within Arivis Pro, there's kind of different types of objects. So there's traces, like what this is considered, and then there's segments. So if I go into this objects table here, and I hit this here, get rid of some of these stored values, traces, track.tracks. Okay, so there's a few different examples of objects types. One is a polyline. So this is kind of like an automatic tracer. You guys have the neuron tracing module, but if, for example, you guys didn't have the neuron tracing module, polyline is kind of the same idea, where it'll find whatever local contrast is from whatever this intensity information of your channel is, and you can then draw it and then get information about the length of this line, angles. You can probably pull out volume of this too. We have also segments. So segments would be what we were looking at Tuesday when we run like a deep learning module to get like objects segmented out. So like these nuclei, for example, or proteins, using like a blob finder or cell pose, those would be considered segments. Traces here are going to be the neuron traces, and the reason being is that traces and segments kind of hold different measurements. So for example, if I have a segment of an object, I'm not going to be able to pull out information like any spine information, no information on neuron volume or structure, I should say, so that we can get things like the length of this, tortuosity, number of branch points. So that's kind of where those nodes would come into play. The length of each of these branches, number of sections. So all of this stuff would kind of be encoded within this trace object. And then we have tracks as the last object type. So tracks would be, to use the same example that I used on Tuesday, was doing like T cells and tumor cells. If you have like a tumor cell and you have a T cell coming in and moving away, and you kind of want to track that, that would all be considered a track. And then you can get things like the speed of this, etc., other information. That being said, you can take these traces and you can convert them to another different object type. So take a trace, convert it to a segment. And this could be useful for maybe you pull out all your information of a neuron, but now you want to use um, some masking. We can't technically mask with a trace, but we can mask with a segment. So you can just take this, if I do it really quick, go into my objects table here. I'm going to select all of my neurites of interest. And then we can go into this objects table. And then you can say, where is it? Right here, convert traces to segments. I'm going to say all of these are going to be converted. And then now these are considered objects. So it's kind of the same thing. It looks just like it. It just has different information. But that being said, I do have, so for example, we can't technically get volume of a mesh. We can go into traces and get volume this way. But it just has a little bit of different information that's kind of compiled within it. Right? But we still have, if when we did that conversion, it kept all of the same like neurites, the traces that we converted them from. So it's not like you're deleting any information, you're just kind of adding on to a layer. Now I'm going to get rid of this segments. We'll bring them back if we have to. Okay. Any questions so far about tracing neurite? Didn't get. Let me hit save. Let me go back on this. So the another option that we can use for tracing. Right now, I use this this threshold based method of tracing. So that's just taking a this intensity information. There's also this probabilistic reconstruction, and this has a lot more flexibility and options. Extend this a little bit. We'll put on this view.
Can I ask a quick question?
Yeah, go ahead.
When would you choose to use the threshold versus the probabilistic? Are there like different types of data you'd use for each?
I would try both and see what works better. Probabilistic, I find in practice, is a little bit more useful for when you have images where there's a lot of diversity within your neuron traces. So, for example, if you have like a neuron, the primary dendrite is probably going to be the largest, and then it's going to go into secondary, tertiary, etc. branches, and typically they get smaller and smaller. Or maybe you have different regions of the neurite that look a little bit different. So maybe you want to get dendrites and an axon, and the axons look completely different from the dendrites. Then you would want to have, in this case, probably probabilistic because you have a little bit more flexibility. So this is a case where you would set your upper and lower limits for branch diameter. So if I had like this axon that I want, which is really large, and then I have these tertiary dendrites that I also want to get that are really tiny, then I would say, you know, whatever that lower limit is, set that lower limit, whatever that upper limit is, set it, and then see how it does.
Okay, thank you.
We can give it a shot here. So this is the its best guess again. And you can see it's kind of pretty much the same, performing the same way. And this also may be, for a lot of the cases, probabilistic and intensity-based tracing may give you the exact same result. There's not necessarily a better or worse way, but I would just play around with it and see kind of what gives you the better result. So let's set something up here. This would be easier if we go now in 2D, because then we can use this ruler tool. to measure our objects. This way we don't have to guess. So I'm gonna go and find, these look pretty small in this area. I think this is kind of one of the smallest ones. And it's already getting it, but let's just be sure. Set this as lower value. Then I think this is probably towards the larger end. And it's missing it. And it's probably missing it because you can see right now my limit is 10.5 and the diameter of this is around 18 or so. Set this as our upper value and hopefully it'll fix it. There we go. Can also do this tubularity sensitivity. Let me go ahead and find. Oh, also another quick tip. If you want to go into that help menu, instead of having to click all of this and looking or going up here and searching it, you can just take your cursor, scroll it over, whatever operator you're interested in looking at. You can hit the F1 button, and that'll give you like a shortcut. So I'm just hitting F1. It brings me right to this neuron tracer. I'm looking for this tubularity metric. So it's looking for these tubular parts of the image. So something that kind of is long and filament-like. And there is a certain level of kind of what that should look like. So I guess the more spread out it is, the less tubular it is, the more confined and straight it is, the more tubular it is. So usually for these, I just start with these calculators because they're usually a really good starting point. And it's going to be based on whatever the ROI is in here. So if I hit this value here, this is like 43. And then if I go and I zoom in up here, this value is going to now change to whatever this my view is in here. So usually I'll just find a nice area that maybe is the software has a little bit of tough time. So maybe if I find it messes up in this region a lot, then I'll use this to keep in focus. Then I'll hit something like this. And then just see how that looks. So it's going to go and calculate a preview. Wait a second. Okay, it doesn't look terrible. So it's recognizing this branch is off. So it's kind of saying, hey, this is underneath that limit of 18.16 that we have set now. And if you remember when we did that threshold based one, it kind of created like a little branch right in here. Let's see if it does it here. So in this case, this is where maybe if you have like some weird structures in your neurons like this that you don't want to necessarily keep. So if you don't want to draw another branch within here, that's also a case where something like this would help. And the reason being is because this diameter is like 30. So it's going to recognize, hey, it shouldn't be that large. So I'm just gonna kind of ignore it and make sense of whatever this 18 micron is. And these values also aren't like super strict. So if you have a part of your neuron that's like, let's say 19 microns, it'll still recognize and be able to pick it up. It does have a little bit of deviation from these values too. Right. And so for here, I think this is kind of looking decent. Let's just see how it looks when we actually run the math on this. So I'm gonna go ahead. We can either hit this run pipeline to here. I'm just gonna hit this, the step forward one, telling us it's running. Give it a second. All right. So we have less results than before. I think we had 9 before with some extra neurites kind of in the background. And let's change this to a random color. So we have an idea of where it's messing up. Go into our 3D viewer. Okay. So it looks like this is all now one trace, except for this beginning part right here. And that's it. So it's just getting this, and I think that's it, right? So there's 2 left. All right. So in this case, maybe probabilistic is a little bit better. And it just depends if you want to kind of use it to get these, if you do want your neuron tracing to get these spines, or if you kind of want to go and say, not that one. Here, correct it there. And also do that. This, I'm wondering if this was because I did not extend my thing all the way. Oh no, that's why. So we can either go in and just adjust our settings again, but looks like it's recognized. Here, we can do something like that. All right. Then let's go a little bit more into our feature table and then look a little bit into the information that we can pull out from this. So just to show you again, have this objects table. It's right next to this analysis panel button. You can also go to objects table up here. We're going to go hit this feature columns. And if you remember, I mentioned that there's different types of objects. So there's segments, there's polylines. In this case, we're going to go back to our traces, get rid of some of these other ones. All right. So let's take a look. So we can get the number of maybe neurites and spines.
Just a quick question.
Yep.
Do we have the center of the neuron here?
Let's see. Can see, it might just say that within this object here, it's going to, you see this bounding box right here? It may just take this bounding box coordinates and then just find whatever the center of this is. So we can actually see what that looks like by changing our object style. Approximation. So we would have to probably convert this to a segment first. So let's do that real quick. And then if we go into our add a style for this, here and here we can get centroid information. Quiet. Let's see. Centroid. I have to do it in 2D. So this, my guess, is where the centroid would be. If I go, yep. So if I hit centroid, so and then approximation. So yeah, it looks like it's just taking the center of this bounding box.
Okay.
But that is for any of the objects, any objects, traces, polylines, anything that we take, all of that information still stored within the software. So may not be useful for this case of a neurite tracing of where the center is, but if you're doing something like maybe like nuclear transport assays where you have like a nuclei and then you want to see like how it moves out to the cytoplasm, you can kind of get like a center point and then track distances. Or if you want to say like, I have this object, I want to see how far proteins are from the center of this object, having information like the center of your objects can be useful for that. And then we will go into this, I think, on Tuesday when we go into colocalization. But one of the things with colocalization, you can also say, give me, or like, if you want to do distance measurements, not quite colocalization, but the same idea. If you're looking relationships between 2 objects, you can say, if I have object A and object B, measure from the surface, so the exterior, so like just on the outside of this object to the other outside of another object. So we can say like from this exterior to this exterior, Or we can say measure from the center of an object to the exterior surface of another object, or you can do center to center measurements too. So this can be useful if you're looking for where objects are in relationship to each other. You have a little bit of flexibility on kind of where you want those measurements to be. Okay. Yeah, so then it'll give you the coordinates on kind of where that is. Let's see, what else can we get? Get the volume of this, and then maybe we can look at the number of branch points. We can look at the max level. See how that looks. Okay. Then we're gonna do just the trace neurite. Let's get rid of this. Get rid of this other volume. Okay. So here, so this is where we're starting from. We just have one object. Oops, where did my table go? We just have one neuron, our neurite, and this is the total volume of this neurite. Again, this objects table is interactive. There's just one object, but if you double click on this, or if you click on this, it'll bring you to its spot within the objects table. There's no cell bodies, which is good because there are none. And then it's saying that there's just one neurite, which is also good because that's what we are looking at here. Maybe if I make this a little smaller. Yeah. We also would get center of neuron coordinates right here. The number of branch points. So go back to here. So these branch points would be regions of larger deviations. So my guess is that it would count this as a branch point, this larger one as a branch point, etc. This one over here, a larger one. Go 3D and look at it too. Or it may just be all of these branch points right here because we have 19, right? Yeah, it looks about 19. Okay. And then the level of this. So there are, in this case, only two levels. So your primary, because this is recognizing as a primary neurite, and then any of these deviations would be the second level. And then there's no further deviations from this. So there's not like a branch from here to here. But wondering if we could kind of force it to say that. You can get other information too. So that's kind of some of the information you can pull out from this. And then again, you can always convert this to segment right afterwards. And then say, give me other information, like intensity information, sphericity. Maybe you're looking at a cell body, and maybe the cell body looks a little bit warped. You're trying to test drug or something. You can kind of get measurements like that. So they can build off of each other. You can get information of it as a segment, and then information of it as a neuron. Right. I'll also mention we can do object styles. I kind of showed you we can just get that different coloring, but we can also change it, make it look glass-like. So this could be useful if you do this. So object style, trace neurite, glass here. There we go. Be something a little, maybe red because that'll contrast. And now you can see now it's transparent. You can see right through it. And this can be useful if you're doing like movies or something, which we'll go over next week. If you're could be useful if you have proteins within your neurons and you kind of want to see them inside while also looking at the segmentation on the outside. Let me see if there's anything else. Okay. All right. So I think that covers neurite tracing. Now we'll go into neuron tracing. So I'll go and save this. And get rid of this for now. Yeah, I don't want to save you. Go to this neuron with cell body. And let me turn this off in 3D because it'll slow down my computer. All right, so this is what we have for, I have for an example 3D data set for a neuron with a cell body. A little bit busier, a lot more background in terms of like maybe neurites that are within this ROI but aren't touching or directly connected to this cell body. Some other stuff in here. So the difference between neuron tracing and neurite tracing is the cell body. So if I go back and I hit this, we'll hit a new pipeline. Again, we're not going to do any segmentation and we're not going to do any image processing, so we can just skip those. Within this, you know what, actually we do need to. So I'll show you really quick though. If we go into this use objects, there's this special, and then there's this neuron tracing. So the what it's asking for is where is your cell body? And we don't have one created right now, so we have to first create that cell body. So this is where we would need to do a segmentation step first. And maybe we'll do blob finder. And the reason we do blob finder, I think it had it up there. I just got rid of it. Let me bring it back. So it detects small rounded objects in dense, noisy environment. So it is looking for objects that have like a circularity spherical component to it, which is what the cell body pretty much is. Okay, so we'll go in 2D, and the first thing blob finder is going to ask for is this diameter. So let's go in 2D, let's find our cell body, and I'm going to find where it's the largest. Like 13 maybe. And remember, this is, I'm measuring in 2D, but this value is going to be, it's a 3D object, right? So even though I'm measuring along this axis, it's saying it's about 16. The diameter of this may be a little bit different in like the Z dimension. So I would pick it as a starting point, but go with whatever value works the best. So a little eyeball right here. Move this to accuracy. And what I want to do is try and do as little filtering as possible after the fact. So I would like to just get the cell body, but then also ignore all these other smaller objects. And it looks like it's not doing that. We can change this probability threshold maybe to higher than 40 maybe. And this probability threshold is going to be kind of the range of the histogram that you're going to be accepting. So we'll let this load for a second, see what it looks like. So think of it as like a more, it's like an intensity threshold, but it's taking a histogram range rather than a single value. That looks a little bit better. You can also use this eyedropper and you can just drag it over a region of interest. And maybe not. There we go. Okay. So you can just take it and kind of drag it, or you can just pick a single point. Either one works. I prefer to just kind of drag it because then it's taking your object of interest is going to be captured a little bit better. Go to this result map. All right, now we'll let it give a second and see what it looks like. I'm hoping that we can get rid of this little bit right here. So, okay, a little bit better. Maybe we reduce it just a smidge. So like maybe 75. Just because I want it to fill around the edges here as best as I can. Maybe just a little bit more would be good. Finishing up. A little better. And then this split sensitivity, we can keep it there for now. All right, let's see how this looks. So I'm gonna hit run pipeline to here and bring this view out. Let's see how large this image is. This is a little bit bigger. It's about 157 megabytes. Might take a second to run. And while this is running, this pipeline is going to now recognize, even though we didn't finish running it, it recognizes that there's a function up here that has an output and an object. So now we can put in for the cell body, we have the option to add in an input. So I'm going to hit this, and it's going to be that blob finder. I didn't call it anything else, but I could have called it the cell body. And then can do, we'd have to wait for this to run to see how it goes. Sometimes this math will, for this percentage it's running, the math is determining what this is, but sometimes it goes slower just because it's doing the math. And then towards the later half of it, it's just doing a rendering. So you can see it's now starting to move a little bit faster. Point being is that it doesn't always just, if it's going a certain speed, there are certain chunks of this pipeline where it may just speed up and finish really quickly, which is what I'm hoping will happen here. And then for this neuron tracing, we also have the option to keep only neurites connected to a cell body. So this would be if you have, I'm not going to switch to it now, but in that 3D viewer, you saw that neuron with the cell body and everything extending, and then you saw these neurites that looked like they weren't from that, from this neuron. So if we don't want to keep those, let's just select this and it'll automatically filter them out. This is finishing up. There we go. All right, so we got some more than one blob, and that is not what we wanted. So one of the things we can do, I'm not going to rerun it just because it took a little bit of time, and I don't want to spend that time again. But what we can do is I would have probably just lowered the split sensitivity, or
I have a quick question.
Yep.
Why don't we just point it, manually point it? Like why we need to go through the broad finder?
Can you repeat the question for we
Can
don't
we
have to
just
do a
manually click like where the cell body is? I mean, I don't think we want to do that in the batch process. I mean, each neuron tracing is an individual project. We
Yeah.
have the 3D, we just click where the cell body is.
We would have to create the cell body, so you can still batch it, and we would just have to kind of fine tune these results a little bit, or we can use that custom cell pose and use custom cell pose to say recognize these cell bodies if we don't want to kind of refine this blob finder. Alternatively, we can also use this. Let's see if we use this magic wand tool, how it looks. So that's kind of like a point and click. We go. Do something like that. And I just do this magic wand, get rid of this blob finder. So that's our result there. So faster.
Yeah. Yeah, I think
So you could do the
this
magic
is
wand too.
okay.
And then we would probably refine this to be, because it looked like it was getting some of that dendrite. So maybe we would just do something like that, maybe. A little bit more restrictive. Get rid of this. Oh, I don't remember. I think it was this one. Right? Yep. And that looks a little bit more. Yep. So you have the option for both.
Okay, thanks.
Yep. So we'll put in this blob finder. Or we can do this. Oh, how do we do this? There's a way that we can take these segments and make it into documents, features. I'm trying to make this available within this here, and you have to just copy it into the pipeline. Otherwise, you'd have to batch it. Let me just see. That I will look into. I know there's a way I've done it before. It says no. I'll look into that. Oh wait, it's right here actually. Now I think about it. It is not. Compartment. Okay, yeah, I'll look into that. I thought copy objects to document. Can do output. I'll look into that. And then, oh yeah, so for getting these blob finders, so just a single kind of neuron from this or cell body from this, let's first find where our objects are in this image. And one way, so what I would do is I would just normally fix up the pipeline to be a little bit more restrictive, but just because I don't want to run it again, let's just do some filtering. So I'll do this, not in the right. We want to do it in our segments. And we want to get rid of these values here, get rid of this neuron volume. Okay. Can do ordering. And now I see this really large object is our one. Oh, it did break this up into different ones. Okay. So let's do this randomly. Okay. So we have these different chunks and we only want the cell body. So one thing we can do, aside from fixing up that pipeline, we can add a filter to this. And so one of the easiest ways to do it is just find your object of interest, right click it, and then hit this add object to feature filter, add object feature filter. And then it's going to add it in right here. It's going to add it in with the value, the feature that we want to filter based off of. And the way that we do that is you just find your feature of interest, find your value, and then right click over that. So if I wanted to filter based on voxel count and get like the smallest one, then I would hover over to the voxel count column, go to the row that I want, and then add it there. Same thing if I want to do it by relative volume, can add it there. And then that'll populate with all the values in there. And then here I can say filtered cell body. Let's do this first because they're both purple. We can actually run this part first. And the input is going to be this blob finder. It's of type anything. So again, segment, trace, track. There's also regions, markers, and groups. So we'll just say it's any of these. Run. I think I little tip, whenever scrolling through this, I use my my mouse like wheel. Sometimes if you go and you're scrolling, depending on where you click, you can actually change the values accidentally. Of course, now I'm not going to do it, but just be careful when you're scrolling. Either just do it over here, just drag and drop. But there we go. We have this now segmented out, but there's this little region, right, that has this little piece right here that we don't want. So easiest way is doing this object splitting. Like an X-Acto knife. You can apply the split, and now you can either do that beforehand or run it and then run another object feature filter. Or just delete it. All right, so we have our cell body here. Let's change this to client, just so we can see it. All right, now we can go and run this neuron tracing. This is the channel that we want. We're going to run it on this now filtered cell body. We're still going to do threshold, right? Yeah, let's start with threshold. Maybe I'll lower this to like 50 to start with. Let's put this eyeball back on and then do 4D preview. Before accuracy. Maybe we'll do like halfway just to speed it up a little bit. So already here, oh, okay. Zoom in a little bit. This. Okay, not bad. Looks like it's missing some spots in here though. So this is a sign. Let's reduce this a little bit. Maybe to like really low. Yeah, like 25. And this should fill up. There we go. The other thing is you can see now it's saying this should be a million little narites, which is definitely not true. So let's select this, but then also raise this value, because this is going to say that the point from like a cell body to a neurite, the smallest value that that'll be is under 2 microns, which is probably why we're getting all of this and why we're also maybe missing some of these regions here. So let's just say it's like 7, something high. And these should, so some of these dropped off, some of these didn't. And raise this accuracy again. These should fall off because we do have this keep only neurites connected selected. I don't think it applies it in this math or in this preview. Let's give it a shot. And then the last step would be, for any pipeline, you're also going to be asked. Okay. So cut rid of that spot. And this is our trace. So again, we had that select keep only neurites connected to a cell body. That's a really easy way of getting rid of all of this. I'm going to call it junk right now. Stuff that we don't want. And then we could probably, and then we also have our cell body, right? So then we can just add this cell body back onto it. So maybe we would do some manual adjustments here, just because it has this little dip. Where'd it go? This little dip right here in intensity that it's probably saying, oh, that's the end of it. So we can either do that or lower this 25 to like maybe 20. Again, it looks like a lot of this background is like right here would be the background. So maybe 20. You know what? It ran really quick. Let's just see if that works. Let's just do that. So I'm picking usually right before that histogram starts to kind of spike up, because that's going to be, I mean, that's going to be your background 9 times out of 10. We'll cut off that background. Let's see how that's looks. I'm hoping that'll fill it in. But it's kind of like if you are using it to kind of fill in some of those lower intensity regions, you can also run the risk of, yeah, got it. Connecting some of those other, no, okay, got it there. Like other spots that we maybe don't want. It's like it's kind of getting it actually now. Look at it. It doesn't look like there's any like large areas that it's really messing up at. Just taking a look here. So again, kind of like here, I probably wouldn't lower this value anymore. And I would just go and trace these myself. Straw. And yeah. Find the right angle. So usually if it has trouble with this, it means the intensity is really low. You can still draw on it. It's just not going to be as snapping like it should. Find it. First. This. Okay, something like that. And then yes, we want to also correct this too. Let's see how this looks really quick. No, that's the end of it. Okay. Right. And then the last thing for any of these pipelines is going to be how you want to save it. So for example, I don't think we need this blob finder that had, so if we go back and we look at this result, remember we had these pieces here that were kind of broken up and cut. We don't want these, right? So we want this result here, this filtered cell body. So when we go down here, we can just say, give me this filtered cell body and give me the results of this neuron tracer. I don't want this blob finder. So that's what I would recommend. Just pick the objects that you want that are necessary for your measurements or your analysis. You don't have to save everything unless you'd want to save it to like look at it and confirm results, or you just like saving everything. And then do that final step. And we have it here. With viewing objects in the objects table and then seeing it in here, you can choose to select just a single type of object. And that's if I just hit the name, it's just going to select that individual thing. Oh, I didn't hit store object. So there we go. Let's run it to here. Okay, now it's saying that is because I am working off of my OneDrive. If I hit here, if I just want to look at the filtered cell body, I would just click the name. But let's say I want to view it with another object overlaid on top of it, so like our trace neurons, then you would hit the boxes here. And then that would kind of add additional layers to this. Okay. Then let's say we get all of our measurements. Let's say I want to get from this. So the segment, right, is going to be that cell body. So maybe I want to get from that segment, that cell body, I want to get the sphericity of it. Let's just get the scaled version. So to the pixel size of the volume. And then let's get the volume of this. Oh, sorry, this would be the volume scaled. And this would be sphericity. I'll actually look at that value. And then from let's just get everything. Maybe not all of them, but we'll do a total length of all of our neurites. Okay. So we have our sphericity and our volume listed here. You can go and click on this. It'll zoom in and show me it. We have our length here. And then I can say, after here, we can go to Excel export. I don't know if I showed this Tuesday, but for here, you can choose all of the features that you want to export. You can just select them here and it'll populate them again. So I have these 4 here, or you can just click here and select more if you want to. You can do all of the objects that are within this here, or just the ones that are selected. So do something like hit selected. We do summary is always nice. It'll keep it as a separate, I think it's just going to be an Excel file, but then a different tab within that Excel file. We didn't do any parent-child relationships. This will be in co-localization. We'll talk about what master detail means and this master detail up here. That'll be Tuesday. But if you do have like objects that are associated with one another, you can kind of have them keep those associations with the tag IDs and names and everything in that Excel file. So this way, when you export this data, you're not just given a whole bunch of values and lists, right? Which you can kind of lose track of. You can say, if I have object A, that's like a nucleus, and it had 100 proteins within it. Give me the Excel file where it says, here's object A of that nucleus, and then all the tag IDs of those children will also be reported. So then you just have to do like a finder or something like that. And you can associate them. And then, all right, so all of this is good. Put it in my demo folder. All right, hit export. And that's what we have here. So you have your objects table, exactly like what you see within here, this table in here. And then we also have our summary here. Okay. And you can have this concatenate it. So I remember I mentioned Tuesday when I'm saying if you have like experiments, like control condition, experiment 1, experiment 2. You can choose to have it all concatenated. So instead of 18 different Excel files, like if there's 6 in each condition, instead of having each one have their own Excel file, you can just say, give me a concatenated file. Just makes it easier so you don't have to search through a ton of stuff. Okay. Trying to think. I think that covers most of neuron tracing. Let me take a look and make sure I'm not missing anything. Hmm. Yeah, we already did split tracings. That's kind of with this structure within here, kind of deleting some nodes. Most of this is really, these are the only 2 functions that you'd be using within Neuron Tracer or um, this Neurite Tracer. You can also do some pre-processing. So let's say, for example, let me find something. I guess here, this is technically connected, but it's just connected tangentially through this branch, not the cell body. But let's say I wanted to actually pull in this and kind of make it out here, like this structure here, this branch, and it's kind of messing up a little bit, right? So what we can do is some pre-processing in this case. If this automatic tracer doesn't work, or you just don't want to, maybe there's like a lot of cases of where you have like these really low intensity regions, then it picks back up again. Instead of having to just manually correct it all, which you could do, you can then try and do some pre-processing and see if that helps the automatic tracer first. So yeah, we'll just restart in a new pipeline. Some image processing. And one of the things I want to point out here, I mean, it would have your typical like processing. So if you want to do some denoising, if you want to do background correction, anything like that, masking, etc. One of the things that I think can be useful is this shape detection. And I'm going to go and find, let me see. Oh, I just got rid of my objects. So this would be, let me find the cell body again. Same thing as before, we'll kind of measure our upper and lower limits. And then you can see it's kind of, yeah, let's make this a little bit. Make this smaller. Pre-processing does take a little bit of time to run. And try and see if it goes. Is this sensitivity? Nope, that is not what I want to do. Let's see how long this will take to run. I'm going to do a preview first, but essentially the point is that it can help to bring out some of these lower intensity regions, and then you're kind of doing like a correction of this by saying there's this upper and lower limit of these filaments. Right here I have selected filaments. You can also use it if you have any spheres or other objects within there. You would pick the same thing. This is also in 3D, so okay. You can see how long this takes. It might take a little bit to run. Yeah. Yes, we can see the step is run, so let's try this now. Yeah, this is going to take too long to run, but this is what I would recommend if there are some cases of where maybe your images like have really low signal to noise ratio and you want to kind of improve it a little bit. I would recommend doing some pre-processing. Can also do some normalization within here. So not that right here. So take maybe something simple, but it would kind of give you just like a single value for everything. It'll normalize to do something simple, or you can do based on the histogram, you can do some normalization there. This is also another way of being able to improve contrast of your objects. So here we're just going to take, I'm going to just kind of show you, but we're just taking the upper limit of this histogram because our objects are going to be pretty go and find like a really dim region. Maybe something here I'm looking at that's really dim and hard to make out. And you can see that you can still see the individual pixels, but now they're just kind of scaling and normalizing them. And again, you can change this value to be a little bit. It's going to reduce it a little bit, some more background, and then maybe you can do then like a denoising after the fact here. You see how long this will take to run. I think this is also going to take a little bit of time. Okay, that's a little faster. And then we can do like a Gaussian filter, or no, maybe median's better. I always like median. Yeah, median always works really well for kind of despeckling stuff. So we can do something like this. Raise the kernel size to be a little bit bigger. Then enter. This accuracy. That shouldn't take long to run either. And a little bit busier, I would you probably want to refine this a little bit more. But you can see at least the point being is that all of these lower intensity regions are kind of normalized now. The only thing you want to make sure that you don't do is oversaturate to the point where, let's say you had two objects that were close together but not touching, and then if you're oversaturating it, now they're going to be touching. That's going to mess it up. But so there's definitely a fine line, but this would also be a case of you don't technically need this intensity information to create the objects, like the raw intensity information. So you can do this normalization, do this denoising, and then once you have your objects traced, remember, we're not changing this data permanently. So that object is going to be built off of this map, but it's going to be, or this mask, but it's going to be applied to your raw data. So if you still want to go back and get your, like, let's say the intensity of these objects, or these traces, you can still get them using the raw data, even though you built your tracing off of your normalized and pre-processed image. Okay. I think that about covers neuron tracing. I think if there's anything else. We also can do, let's say, so if anybody, I know some people like Shoal analysis. We don't have Shoal analysis. Yep. The question?
I would just feel it's kind of, I think it's enough for today. I think it's a lot of things.
Yeah, we can save the last half hour then for another day. And we'll kind of add it on to another day. But I think that kind of covers the rest of neuron tracing. There's not a really, it's just doing more pre-processing we can go into, but the steps are going to be the same, whatever gives you the best result. And then we would also work with you to improve, if you have questions on how to, you can't get a tracing done really well, we can work with you to make sure we can get something.
Okay.
Stop sharing. All right, does anybody have any final questions before we wrap this up? Right. Let me go ahead and stop this recording.